Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about carbohydrate. So let us start learning of carbohydrates with classification of carbohydrate. How we will classify carbohydrate. So carbohydrates and biochemistry. I will focus on this and then we will move. You know, my friend, carbohydrates are compounds of tremendous biological importance. They provide energy through oxidation. They supply carbon for the synthesis of cell components. They serve as a form of stored chemical energy. They form part of the structures of some cells and tissue. Carbohydrates, along with lipids, proteins, nucleic acid, and other compounds, are known as biomolecules because they are closely associated with living organisms. Now, carbohydrates or saccharides, saccharo is a Greek for sugar, are polyhydroxyaldehydes or ketones or substances that yield such compounds on hydrolysis. You can see, my friend, here the structure of fructose. This is the secondary alcohol 1, 2, 3, and first one and the last one in fructose is the primary alcohol, and this is the functional group of ketone. Now here first one aldehydic group and 1, 2, 3, 4 secondary alcohol and 1 primary alcohol. Now ring structure of beta D glucose. You can see this is the 1 and this is the 2. So 1 and 2, this is the down, that is the beta D glucose. You know, my friend, carbohydrates include not only sugar but also the starches that we find in food such as bread, pasta and rice. You can see in this picture. The term carbohydrates comes from the observation that when you heat sugars, you get carbon and water, hence hydrate of carbon. Now, how to classify the carbon or classes of carbohydrates. Monosaccharide contains a single polyhydroxyaldehyde or ketone unit, glucose or fructose. Disaccharide consists of two monosaccharides units linked together by a covalent bond. Example, sucrose. Oligosaccharide contains from 3 to 10 monosaccharide unit. Example, raffinose. And you know, my friend, polysaccharide. Polysaccharide contain very long chains of hundreds or thousands of monosaccharide units, which may be either in a straight or branch chains, cellulose, glycogen, and starch. You can see. Now the stereochemistry of carbohydrate. Two forms of glyceraldehyde. Glyceraldehyde, the simplest carbohydrate, exists in two isomeric forms that are mirror image of each other. You can see, I told you 3 functional group is present. 2 functional group is the same nature for the secondary alcohol and primary alcohol and aldehyde. So this is present in this structure. So this is the first compound belongs to carbohydrate. So these compounds have a chiral center. and this is the mirror image of this glyceraldehyde. So L-glyceraldehyde and D-glyceraldehyde. Now stereoisomers. You know, my friend, carbohydrate contains a number of stereocenter, and these form are stereoisomers of each other. Glyceraldehyde is a chiral molecule. It cannot be superimposed on its mirror image. The two mirror image forms of glyceraldehyde are enantiomers of each other. So L-glyceraldehyde and D-glyceraldehyde. Now, chirality and handness. What does it mean? Chiral molecules have the same relationship to each other that your left and right hand have when reflected in a mirror. If you want to practice it, take the mirror and your hand is with you and practice it. You can see, I look at this picture, mirror and right hand, where what is changing here. Now, chiral carbons. Chiral objects cannot be superimposed on their mirror image. Example, hands, gloves and shoes. A chiral object can be superimposed on the mirror image. Example, drinking glass, spheres and cubes. Now, any carbon atom which is connected to 4 different groups will be chiral and will have 2 non-superimposable mirror images, and it is chiral carbon or a center of chirality. If any of the 2 groups on the carbon are the same, the carbon atom cannot be chiral. Many organic compounds, including carbohydrates, contains more than one chiral carbon. So, example of chiral carbon atoms you can identify in these examples. Example of chiral carbons in carbohydrates. Identify the chiral carbon in the following carbohydrates. Now you can see the erythrose, you have glucose. Now you know, if you choose this carbon, this is the different, this is different, this hydrogen is different, this is the different. If you choose this one, this group is different, this group is different, this is the different, this is the different. Now, second rule. When a molecule has more than one chiral carbon, each carbon can possibly be arranged in either the right hand or left hand form. Thus, if there are n chiral carbons, there are 2 to the power n possible stereoisomers. So, maximum number of possible stereoisomers 2 to the power n. You can see here 1, so 2 per power 1, 2 possible isomers. If you have the 2, you can see this carbon, this group, this group, this group, this group, this group, this group, this group. So for this carbon, this different. For this carbon, this group, this group, this group, this is different. So there are 2 chiral center here. 2 to the power 2 is equal to 4 possible stereoisomers. So number of stereoisomers. What is the maximum number of possible stereoisomers of the following compounds? You can write it down and practice it. What is the maximum number of possible stereoisomers of the following compound? That is the cholesterol. So here you can see 2 to the power 8. Now you can look at here. This group, this group, this group is different. This one group is different for this carbon. Here this one different to this carbon, and so on. So 2 to the power 8. 1, 2, 3, 4, 5, 6, 7, 8. So cholesterol have 256 possible stereoisomers, but nature make only one. Now Fischer projections, what does it mean? You know, my friend, Fischer projection, this is a projections, are a convenient way to represent mirror image in 2 dimensions. Place the carbonyl groups at or near the top and the last chiral, a chiral CH2OH at the bottom. You can see. So left and right. So L glyceraldehyde and D-glyceraldehyde. Now naming stereoisomers. When there is more than one chiral center in a carbohydrate, look at the chiral carbon farthest from the carbonyl group. If the hydroxy group points to right when the carbonyl is up, it is the D isomers. When the hydroxy groups point to the left, it is the L isomers. So you can see L erythrose and D erythrose. Example of Fischer projections. Draw Fischer projection of D and L lactic acid. Give the structure of D-glucose, draw the structure of L-glucose. You can practice it. Identify the following compounds as D or L isomers and draw their mirror image. You can see which one is D, which one is L. Now, what's so great about chiral molecules? Molecules which are enantiomers of each other have exactly the same physical properties: melting point, boiling point, index of refraction, but not their interaction with the polarized light. Polarized light vibrates only in the one plane. It results from passing light through a polarizing filter. You can see ordinary light vibrates in all planes. Unpolarized lights, then you can put the polarized lens. It passes through the plane of polarized light. You can see the tube containing solution of enantiomers, and these directions, this is the PPL, plane polarized light, only two directions. Now some terminology related to optical activity. A levorotatory substance rotates polarized light to the left, so L-glucose or minus glucose. A dextrorotatory positive substance rotates polarized light to the right, D-glucose or plus glucose. Molecules which rotate the plane of polarized light are optically active. Many biologically important molecules are chiral and optically active. Often living system contains only one of the possible stereochemical forms of a compound, or they are found in separate systems. L-lactic acid is found in living muscles. D-lactic acid is present in sour milk. In some cases, one form of molecule is beneficial and enantiomers is another form is the enantiomers is poisonous. Example, thalidomide. And humans can metabolize D monosaccharides but not L isomers. Only L amino acid are used in proteins synthesis. Now my friends, monosaccharides. Classification of monosaccharides. The monosaccharides are simplest of the carbohydrate since they contains only one polyhydroxyaldehyde or ketone unit. So monosaccharides are classified according to the number of carbon atom. They contains number of carbon 3, class of monosaccharide triose, E4, then tetrose, 5 pentose and 6 hexose. The presence of an aldehyde is indicated by the prefix aldo and ketone by the keto. Classification of monosaccharide. Glucose is an aldohexose, aldehyde plus 6 carbons, and ribulose is a ketopentose, ketone plus 5 carbon. You can see the glucose and aldohexose, and ribulose is a ketopentose. So classify monosaccharide. You have the molecules, you can classify it. Now family of D-aldoses. L form not shown here. So D-glyceraldehyde, aldotriose 2 to the power 1, 2 isomers. D-erythrose and D-threose, aldotetroses 2 to the power 2, 4. And same aldopentoses 2 to the power 3, 8. Now you can see the D-allose, D-altrose 2 to the power 4 is equal to 16 possible isomers. The family of the ketosis, L form not shown here, this dihydroxyacetone, dihydroxyacetone, ketotriose 2 to the power 0, 1, and D-erythrulose 2 to the power 2 isomers, and 2 to the power 2, 4, that is the D-ribose and D-xylose. The family of the ketosis here like D-fructose and 8 other ketohexoses. Now my friends, physical properties of monosaccharide. Most monosaccharide have sweet taste. Fructose is sweetest, 73% sweeter than sucrose. They are solid at room temperature. They are extremely soluble in water. Despite their high molecular weight, the presence of large number of OH groups make the monosaccharides much more water soluble than most molecules of similar molecular weight. Glucose can dissolve in minute amount of water to make a syrup, 1 gram 1 ml water. My friend, physical properties of monosaccharide you can see in this table. The relative sweetness of sugar. Sucrose, if you have the scale 1, then lactose sweetness 0.16. That type is the disaccharide. Galactose, you can see 0.22, that is the monosaccharide. Maltose 0.32, disaccharide. Xylose 0.40, monosaccharide. Glucose 0.74, monosaccharide. Invert sugar 1.30, that is the mixture of glucose and fructose. That is important. You can see the one scale. Sucrose exactly 1.00 disaccharide and fructose 1.73, that is the monosaccharide. Now chemical properties of monosaccharide. You know my friends, the monosaccharide do not usually exist in solution in their open chain forms, and alcohol groups can add into the carbonyl group in the same molecule to form a pyranose ring. Keep in your mind, alcohol and carbonyl, same molecules to form pyranose ring containing a stable cyclic hemiacetal or hemiketal. You can see a pyranose ring, that is the beta D glucose, beta up, cyclic hemiacetal, alpha down, alpha D glucose. So glucose anomers, what does it mean? This is the Haworth structure, 1 alpha D glucose 36% and beta D glucose 64% and D glucose 0.02%. So there are 3 forms in changing. So this is the your positions 1 here. This is the position 1. So in the pyranose form of glucose, the carbon 1 is chiral, and thus 2 stereoisomers are possible. One in which the OH group point down, alpha hydroxy groups, and one in which the OH group point up, beta hydroxy. These forms are anomers of each other, and carbon 1 is called anomeric carbon. Now fructose anomers. Fructose closes one itself to form a furanose ring. You can see a furanose ring, D fructose, alpha hydroxy, alpha D fructose, beta hydroxy, beta D fructose. Now, drawing furanose and pyranose ring. Monosaccharides are often represented using the Haworth structure shown here for furanose and pyranose ring. The remaining OH group on the ring point up or down depending on the identity of the sugar. Always above ring for the disaccharides. CH2OH always up. So furanose ring and pyranose ring. Examples with friends like these who needs enomers. You can see. Identify the structure below as B, the alpha or beta form, and draw the structure of their enomers. Now, oxidation of monosaccharide. Aldehydes and ketones that have an OH groups on the carbon next to the carbonyl carbon groups react with a basic solution of copper 2+. Benedict reagents to form a red-orange precipitate of copper 1 oxide. Now, sugar that undergo this reaction are called reducing sugar. All of monosaccharide are reducing sugar. So, reducing sugar, copper 2+ deep blue solution, it gave oxidations product and cuprous oxide red-orange precipitate. You can see the Benedict's reagent blue color, it converted into the red-orange precipitate copper 1 oxide. Now formation of phosphate ester, glucose 6-phosphate and fructose 6-phosphate. You know phosphate ester can form at the carbon 6 carbon of aldohexose and aldoketosis. Phosphate ester of monosaccharide are found in the sugar phosphate backbone of DNA and RNA in ATP, adenosine triphosphate, and as intermediate in the metabolism of carbohydrate in the body. Now, glycoside formations. The hemiacetal and hemicatal forms of monosaccharide can react with alcohol to form acetal and ketal, structures called glycosides. The new carbon-oxygen bond is called glycosidic linkage. So, you can see you have alpha D glucose reacts with methanol. It gives methyl alpha D glycopyranoside and methyl beta D glycopyranoside. Now, glycoside formations. Once the glycoside is formed, the ring can no longer open up to the open chain form. Glycosides therefore are not the reducing sugar. Once the glycoside is formed, the ring can no longer open up for the open chain form. Glycosides therefore are not reducing sugar. Glycoside copper 2+, not reaction. So how you will make the glycoside? You can see, focus on this. This is the glycosides. This OH converted into CH3, OCH3. Now example, glycoside formation. You can see, identify the glycosidic linkage in each of the following molecules. You can see here. Now, important monosaccharides. Beta D ribose forms the sugar backbone of ribonucleic acid, and beta D deoxyribose forms the sugar backbone of deoxyribonucleic acid, DNA, and beta D galactose incorporated with the glucose into lactose, milk, sugar. Now, important monosaccharide. You can see beta D glucose, also known as dextrose and blood sugar. Beta D glucose, also known as dextrose and blood sugar, present in honey and fruits. Glucose is metabolized in the body for energy. Other sugar absorbed into the body must be converted to glucose by the liver. Beta D fructose, also known as levulose and fruit sugar. Fructose is the sweetest of the monosaccharide. It is present in 1:1 ratio with the glucose, fructose and corn syrup. It is often used to sweeten foods since less fructose is needed to achieve the same degree of sweetness. Disaccharide and oligosaccharides. You know, my friends, disaccharide, two monosaccharide can be linked together through glycosidic linkage to form disaccharide. You can see here alpha D glucose and alpha D glucose combine together to forming maltose. Cyclic hemiacetal, you can see alpha 1,4 glycosidic linkage. Alpha 1,4 alpha 1,4 1 2 3 4 alpha 1,4, you can see alpha 1,4 glycosidic linkage. This is the cyclic hemiacetal. Now disaccharide can be hydrolyzed into their monosaccharide building blocks by boiling them with dilute acid or reacting them with the appropriate enzymes. Disaccharide that contains hemiacetal groups are reducing sugar. So maltose plus water, it gives D-glucose and D-fructose. This is the maltose and this is the react with the Benedict reagents. You can see this is react with the reducing Benedict reagents. So disaccharide that contains hemiacetal groups are reducing sugar. Now important disaccharides. You can see the maltose, alpha D glucose, this is known as malt sugar. It is produced in germinating grains such as barley, as starch is broken down during the malting and is formed during the hydrolysis of starch to glucose during the digestion. Lactose, also known as milk sugar. Lactose contains 5% of cow's milk and 7% of human milk. It is digested by the enzyme lactase. Pure lactose is found in whey and watery byproduct of cheese products. So you can see the structure: alpha D glucose, beta D galactose and beta 1,4 glycosidic linkage. Beta 1,4 1 2 3 4 beta 1 to 4 glycosidic linkage. Now important disaccharides. Sucrose, also known as table sugar. Both anomeric carbon of glucose and fructose are tied together in the glycosidic linkage. Thus neither ring can open and sucrose is not a reducing sugar. Sucrose is found in fruits, nectar, sugar cane and sugar beets. Maples syrup contains 65% sucrose with glucose and fructose present as well. So caramel is the solid residue formed from heating sucrose. A flavoring agent called invert sugar is produced by hydrolysis of sucrose under acidic condition, which break it apart into glucose and fructose. Invert sugar is sweeter than sucrose because of fructose. Some of the sugar found in honey is formed in this fashion. Invert sugar is also produced in jams and jellies prepared from acid-containing fruits. Now, oligosaccharides. Oligosaccharides contain from 3 to 10 monosaccharides units. You can see the beta D galactose, beta D glucose, and beta D fructose. Now this is the raffinose, an oligosaccharide found in peas, beans, largely undigested until researching an intestinal flora in the large intestine, releasing hydrogen, carbon dioxide, and methane. This is the raffinose. Now polysaccharides. So I am not here on focus because this is not in syllabus, but you should know for the competitions. Polysaccharide contain hundreds and thousands of carbohydrate units. Polysaccharides are not reducing sugar, since the anomeric carbon are connected through glycosidic linkage. Anomeric carbon are connected through the glycosidic linkage. We will consider three kinds of polysaccharide, all of which are polymers of glucose: starch, glycogen, and cellulose. Starch, glycogen, and cellulose are not reducing sugar. Starch is a polymer consists of D-glucose unit. Starch and other glucose polymer are usually insoluble in water because of high molecular weight. Because they contain large number of OH groups, some starches can form thick colloidal dispersion when heated in water, and flour of or starch used as a thickening agent in gravies or sauces. There are two forms of starch, amylose and amylopectin. You know, starch amylose. Amylose consists of long unbranched chain of glucose from 1000 to 2000 molecules connected by alpha-1,4 glycosidic linkage. Alpha-1,4 glycosidic linkage, amylose. 10% to 20% of starch in plant is in this form. The amylose chain is flexible enough to allow the molecules to twist into the shape of helix. Because it packs more tightly, it is slower to digest than other starches. So starch, you know, amylose helical can trap molecules of iodine, forming a characteristic deep blue-purple color. Iodine is often used as a test for the presence of starch. So, you know, starch amylopectin. Amylopectin consists of long chain of glucose up to 10 to the power 5 molecules connected by alpha-1,4 glycosidic linkage with alpha-1,6 branch every 24 to 30 glucose unit along the chain. So 80 to 90% of starch in plant is in this form. You can see 1 to 6 branches and 1 to 4 glycosidic linkage. That is the branch point. So 1 to 6, that is the 6th position, this is the 1 position. 1 position and 6 position of this. And this is the 1 positions and that is the 4th position. The 1 and 4th. 1 and 6. Now glycogen. You know, glycogen, also known as animal starch, is structurally similar to the amylopectin. So, you know, glycogen, also known as animal starch, is structurally similar to amylopectin, containing both alpha-1,4 glycosidic linkage and alpha-1,6 branch point. Glycogen is even more highly branched, with branch occurring every 8 to 12 glucose unit. You can see here. Glycogen is abundant in the liver and muscles. On hydrolysis, it forms D-glucose, which maintains normal blood sugar level and provides energy. Starches plant amylose, amylose pectin, amylose unbranched, amylopectin branch. glycogen animals highly branched. Now cellulose is a polymer, consists of long unbranched chain of D-glucose connected by beta-1,4 glycosidic linkage. It may contain from 300 to 3000 glucose unit in one molecule. You can see the beta-1,4 glycosidic linkage. Beta, this one is up, beta-1,4 glycosidic linkage. Beta-1,4. First you see the beta-1,4 glycosidic linkage. Now cellulose. Because of the beta linkages, cellulose has a different overall shape from amylose, forming extended state chain which hydrogens bond to each other, resulting in a very rigid structure. Cellulose is the most important structural polysaccharide and is the single most abundant organic compound on earth. It is the material in the plant cell wall that provide strength and rigidity. Wood is 50% cellulose. You can see the plant cell. So most animals lack the enzymes needed to digest cellulose, but it doesn't provide roughages dietary fibers to stimulate the contraction of the intestine and help pass food through the digestive system. Some animals such as cows, sheep and goats, ruminants process cellulose using colonies of bacteria in the digestive system which are capable of breaking down cellulose in a series of stomaches to give cellulose a longer time to digest. Some other animals have longer intestinal tract, example horses, and other reprocess digestive food, rabbits, to allow more time for the breakdown of cellulose to occur. So cellulose is important industrially from its presence in wood, paper, cotton, cellophane, rayon, linen, nitrocellulose, gun cotton, photographic film, cellulose acetate. Now, nitrocellulose and celluloids and rayon. So from here you can see the gun cotton is cotton which have been treated with a mixture of nitric and sulfuric acid. It was discovered by the Christian Frederick in 1845 when he used his wife cotton apron to wipe up a mixture of nitric and sulfuric acid in his kitchen, which vanishes in a flash of flame when it dried out over a fire. He attempted to market it as a smokeless powder, but it is combusted so readily it was dangerous to handle. Eventually, its use was replaced by cordite, a mixture of nitrocellulose, nitroglycerins, and petroleum jelly, which could be extruded into cord. Celluloids was the first synthesis plastic made by combining partially nitrated cellulose with a alcohol and ether and adding camphor to make it softer and more malleable. It was used in manufacturing synthetic billiards balls as a replacement for ivory. Photographic film. It was eventually replaced by less flammable plastic. Rayon. It consists partially nitrated cellulose mixed with solvent and extruded through small holes, allowing solvent to evaporate. Rayon was a sensation when introduced since it was a good substitute for silk, but it was still highly flammable. Now, dietary fiber. Dietary fibers consist of complex carbohydrates such as cellulose and other substances that make up the cell wall and structural part of plants. Good source of dietary fiber include cereals, grains, oatmeal, fresh fruits and vegetables and grains product. Soluble fibers such as pectin has a lower molecular weight and more water soluble. Soluble fiber taps carbohydrates slow their digestions and absorptions, thereby leveling out blood sugar level during the day. Soluble fiber also help to lower cholesterol level by binding dietary cholesterol. Insoluble fibers such as cellulose provides bulk to the stool, which help the body to eliminate solid waste. Now chitin. Chitin is a polymer of N-acetyl glucosamine, chitin, an amide derivative of amino sugar, glucosamine, in which one of the OH group is converted to an amine, NH2 group. The polymer is extremely strong because of the increased hydrogen bonding provided by amide group, chitin. You can see chitin is the main component of cell wall of fungi. The exoskeleton of arthropods such as crustaceans and insects and break of the cephalopods. The chitin is often embedded in either a protein matrix or in calcium carbonate crystals. Since this matrix cannot expand easily, it must be shed by the molting as animal grow. You can see in this picture. Now how sweet it is. Sugar substitutes. This is the saccharin. You can see saccharin, amide and S double bond S double sulfone. The first of the artificial sweetener, saccharin, is the non-caloric and about 500 times sweeter than sugar. It was discovered in 1879 by containing Fahrenberg and chemistry student at Johns Hopkins University working for Era MNC. He noticed that the bread he was eating was usually sweet and went back to his lab bench and tested all the compounds he had been working with that day to find the compounds responsible. It was marketed commercially as a non-nutritive sweetener very quickly, especially for use by diabetics. It was banned in some areas for some time because it was a suspected carcinogen. Aspartame. You can see aspartame. Aspartame NutraSweets is about 116 times sweeter than sugar. It is composed of amino acid, aspartic acid, and phenylalanine, neither of which has a sweeter taste. It was discovered at Surrey by James Slater in 1965, who was preparing intermediate for the synthesis of tetrapeptidate for a anti-ulcer project. Shetler had spilled some of the dipeptide intermediate on his hand and noticed later that there was a strong sweet taste. He went back to his branch and tested the dipeptide and found that its intent was extremely sweet. Aspartame is sensitive to heat, so it cannot be used in cooking food, and it decomposes slowly in liquids, reducing their shelf life. So next is cyclamates, discovered by Michael Sweder in 1937, who noticed that a cigar that he was smoking in lab tasted especially sweet. It is about 30 to 50 times sweeter than sugar. It is often used in combination with the other artificial sweetener. It is heat stable. Cyclamate was banned by the FDA in 1971, although it is still used in the other countries. And you can see the others. Now, sucralose, a non-caloric artificial sweetener approved by the FDA in 1998. The glucose is sucrose is replaced by a galactose, and three of the OH group are replaced by the chlorine atoms. It is about 600 times sweeter than sucrose and is non-caloric and heat stable. Sorbitol, a sugar alcohol, incompletely absorbed during the digestion and contributes fewer calories than carbohydrate found naturally in the fruit, used in commercially in sugar-free candies, cookies, and chewing gums. Now you can see the ST-boat is a plant in the sunflower family, widely grown for its sweet leaves. The leaves contains a compound called STBOL, which is attached via glycosidic linkage to glucose molecules to form ST-bols. That is 250 to 300 times sweeter than sugar, and rebutadiose arabiana contains an additional glucose unit 350 to 450 times sweeter than sugar. The FDA put ribodioside A on GRAS list in 2019. You can see the structure. The Maillard reactions. The Maillard reaction occurs between foods containing carbohydrates and protein under the heating. It resulting in a complex mixture of products and the development of brown color. In addition, many complex flavors are developed. So, it is observing in the grilling or browning of meat, the formation of crush on baked bread, toast and pretzels, the toasting of the marshmallows and frying of potatoes and onion, the boiling of maple syrup, the brewing of the beer, melting of barley, and roasting of the coffee and cocoa bean, the roasting of nuts and other sources. There are hundreds of compounds producing during these reactions, not many of which are well characterized. So this is the end. My friends, I hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.